So thank you for being here. This is Dawn Merritt with the Isaac Walton League of America. We are delighted to be hosting today's webinar. As I mentioned to folks who had joined early, you are in listen-only mode, so if you have questions, please use the question box in your toolbar. So welcome to our webinar, Long-Term Look at Changes in the Quality of Our Nation's Rivers and Streams. It's part of the Water Monitoring webinar series. And I am pleased to introduce our presenter, Lori Sprague, who is a hydrologist with the U.S. Geological Survey in Denver. She is the coordinator of surface water trend studies in the USGS National Water-Quality Assessment project, which collects information on water chemistry and aquatic life to provide science-based insights on surface and groundwater issues throughout the U.S. One of the major goals of the project is to track long-term changes in the nation's stream quality. And they recently completed a comprehensive evaluation of changes in stream quality using monitoring data collected by USGS and 73 other organizations between 1972, when the Clean Water Act was passed, and 2012. So now, for more details on changes in the quality of our nation's rivers and streams, here is Lori Sprague.
Thanks, everyone, and thank you for joining us. And this, I think, is a danger of doing a virtual webinar. Dawn was kind enough to ask, because we'd never met in person, how to pronounce my name, and I think I might have written it phonetically wrong. So it's actually Lori Sprague, and I'm excited to be here today to be able to tell you about some new results that we have coming out of the U.S. Geological Survey's National Water Quality Program. The work was done by our National Water Quality Assessment project, which is part of the National Water Quality Program. And the NAWQA project has been around since the early 90s. And since its inception, we've had three major goals for the project. The first has been to look at status, what is the current quality of the nation's surface water and groundwater. The second objective was to look at trends. So is water quality getting better or worse over time? And then the third objective was understanding. So figuring out what the major natural and human factors are that are controlling water quality. What I'm going to be talking about today is the second objective of looking at trends. We're now 20+ years into the program. We've been collecting monitoring data for a good while now, and we thought it was the right time to stop and say this is when we're going to really focus on looking at trends in water quality. Over time, our funding has declined somewhat, and we've had changes to our monitoring network. And so when we took a look at our network, we said, well, there are some really important gaps that we'd like to be able to fill in geographically if possible. And we knew there was a lot of really valuable long-term monitoring that was going on through other monitoring organizations across the United States. So we wanted to not only look at our own data, but broaden the assessment to include all of that other valuable data. So in the end, we have what we believe is the largest ever evaluation of trends in U.S. stream quality between the passage of the Clean Water Act in 1972 and 2012. When all was said and done, we had over 20,000 trend results, and those covered 51 different chemicals and 38 measures of aquatic life. We started with a pool of nearly 500,000 sites, but when we screened them down, and I'm going to talk a little bit about that, we ended up with about 1,400 sites where we had at least one trend result for one chemical or one aquatic life metric for one of our time periods. So as I mentioned, and as Dawn mentioned, this study is looking at data from USGS and 73 other monitoring organizations from across the United States. We have an online interactive mapper that provides user-friendly access to the trend results, and I will give you a quick demo of that mapper toward the end of the presentation. And the results from this study are going to be a foundation for answering some critical interpretive questions about the causes and the effects of changes in stream quality in the United States. And I'll tell you a little bit about our ongoing efforts on that side of things at the end of the presentation. The scope of our study included a variety of chemical constituents. So we looked at nutrients, pesticides, sediments, carbon, salinity, and then a variety of aquatic ecology metrics, including fish, macroinvertebrates, and algae. We have 4 time periods that we looked at, starting with the passage of the Clean Water Act in 1972, and then moving decadally ahead. So 1972, 1982, 1992, and 2002, all ending in 2012. In order to be as inclusive as possible with our data screening, we went to the U.S. Geological Survey's National Water Information System database for data. We went to EPA Storeit database for data, and then because we knew that there were major monitoring agencies that had additional data that they weren't putting into Storeit, we reached out to a number of other federal, state, and local agencies to ask for additional water quality data that might not be in NWIS or in Storeit. And so the final universe of data that we included in our screening process had 185 million water quality records from almost 500,000 sites and over 600 different monitoring organizations. I want to start by telling you a little bit about how we processed and screened our data so you can understand how we arrived at the final trend sites. We had three major steps in the process. The first was harmonizing all of that data that we got and addressing metadata gaps. The second was making sure that we had the right kind of data over our target trend periods to do a successful trend analysis. And then we wanted to make sure that we had Streamflow data to help inform the analysis and the interpretation of the trends. And I'm going to talk about each one of these in a little more detail. I'll illustrate the process that we went through using an example. So this is now showing you every site in the United States from that aggregated database that we had where there was at least one orthophosphate sample. So we started out with over 100,000 sites that were in consideration for inclusion in trend analysis. And of course, from there, we had to look at how much data was present at each of those sites. The next thing we did was look for where we had complete metadata. So where did we have enough information about the sample to unambiguously know what the data values were so that we could do trend analysis at that site. And this was actually by far the most time-consuming part of the processing for us. It took us several years to do this for all of the data that we had compiled from the 608 organizations that we looked at. I want to tell you a little bit more about that. For the work that we were doing, the key metadata elements that we were looking for were parameter name, information on whether or not the sample was filtered, whether or not the chemical form was reported as molecular or elemental, what the units were, whether there were remark codes about laboratory censoring or contamination that we needed to be aware of, and then a clear numerical value. And I'll show you what I mean by these. Starting with parameter name. So just now thinking of nutrients. We had 10 nutrients that we were considering including in the trend analysis. And for those 10 nutrients, we had over 1,000 unique names across these 608 organizations. So we had to manually map those 1,046 names to one of the 10 nutrients. So we were able to successfully identify 931 of them, but there were 115 names where we just could not figure out what chemical is being represented. Now, this is a situation where we know that the originating data producer, the agency that produced the data, we know they know exactly what it is. The challenge comes in when we combine their data with data from all of these other organizations that are doing things differently. So it's that lack of standardization that makes it difficult to combine all of this data together. As an example, you'll see on here, for orthophosphate, we had 147 different naming conventions, and for ammonia, for example, we had 141. Here's an example of what I'm talking about. So here's an agency that reported inorganic nitrogen, and they specified it as ammonia plus nitrite plus nitrate. We had another organization who reported inorganic nitrogen as just nitrate and nitrites. So these are both very clear. We can work with these. We got a number of other organizations who reported inorganic nitrogen without the specific information on which nitrogen species were included. So the fact that we had two organizations that were reporting inorganic nitrogen clearly yet differently meant that when we didn't get additional detail on what was included in inorganic nitrogen, we weren't able to use the data. And again, that's not because there was a problem with the originating data, it's because there was inconsistency introduced by combining data from different organizations. Another thing here you'll notice, these last 4 rows, that's all from the same organization. The fact that they are reporting them differently suggests that there's some meaningful difference between the data reported under those 4 parameter names, but we can't tell by looking at the parameter name what the difference is. So that's another situation where we weren't able to confidently use the data. The next thing we looked for was information on whether the sample was filtered or not. So whether we were looking at just the aqueous fraction of a chemical or whether we were looking at both the particulate and the aqueous fraction. And so in a given water sample, it is possible to determine both filtered and unfiltered variations of the same chemical, like total nitrogen and total dissolved nitrogen. And whether or not the sample is filtered can make a big difference in the resulting value. And you can't always tell by looking at the laboratory method what the filtration status is, because a lot of organizations filter in the field before the sample gets to the laboratory. So here's an example of the difference that it can make. These are samples that were collected from various rivers in the Northeast, and the same river sample was analyzed in a filtered state and in an unfiltered state. And you can see that, as you might expect, the unfiltered sample concentrations were by and large higher than their filtered counterparts. So it matters whether or not the sample is filtered or not. And in particular for trend analysis, this is an issue because if you have an agency that filtered for 10 years and then they stopped filtering and it had unfiltered samples for the next 10 years, you can see that that might introduce an artificial trend in the data that we might incorrectly attribute to an environmental change. And of course, there are going to be situations where it doesn't matter whether it's filtered or not, that those are indistinguishable because the particulate component of that chemical is so low that it doesn't matter. So there will be cases where it matters and cases where it doesn't, and you can't universally assume any one condition. And so that's the reason in the metadata that we look for information on filtration status. Another big issue that we ran into with the metadata was chemical form. There are things like nitrate that can be reported 2 ways. One is in the molecular form as nitrate, and one is in the elemental form as nitrogen. And these 2, for the same sample and the same value, will differ by a factor of 4.5. So let me show you an example of that. These are nitrate concentrations from a creek in Pennsylvania, and you'll see they're reported nitrate as nitrate. Now you can divide the values reported here by that scaling factor of 4.5 to get nitrate as nitrogen, and you see it makes a big difference. If you think about this in a regulatory context, the EPA maximum contaminant level is 10 milligrams per liter of nitrate as nitrogen. So you want to compare that to the purple values here. But if you had the black data set and you didn't know whether it was molecular or elemental and you made the wrong assumption and you assumed that those black values were in fact nitrate as nitrogen and you compared them to the maximum contaminant level, you would reach an incorrect conclusion about the impairment level of that creek. One more thing I wanted to point out about about metadata is remark codes and the importance of remark codes in data analysis like trend analysis. In the combined agency data set that we had, there were 587 unique remark codes. A lot of them weren't defined or they were ambiguously defined, so it was hard for us to figure out what they meant. And again, this is because of these data being combined with data from other organizations and some ambiguity being introduced that way. In particular, there were 63 unique remark codes that indicated laboratory censoring, and knowing that is very important for a statistical analysis like trend analysis because misidentification of censored data can lead to potential bias in the result. Also importantly, of the 488 sources for which we had nutrient data, 118 of them did not provide any records at all with censored remark codes. You know, if it's an organization that's giving us 5 samples, it's plausible for sure that they just wouldn't have any very low concentrations that couldn't be detected by the laboratory. But if it's an entire state and it's tens of thousands of samples, it's pretty unlikely that they would not have any low concentrations that they've sampled that were unable to be quantified by the lab. So we know that there are some organizations out there that feel like sensor data maybe is poor quality data, and they deliberately withhold it from public databases. So that's a situation where we didn't think that we could confidently use the data for something like trend analysis. This is just going to illustrate the importance of having that information on laboratory censoring. This is also showing nitrate concentrations now for a river in Oklahoma, and these are all of the uncensored values that were present in that data set. If you take the mean of those values, you get a concentration of 0.593 milligrams per liter. Now, if you include those censored values that represent the lowest concentration situations for that river, the low concentrations that can't be definitively quantified by the laboratory, and you include those in the estimate of the mean, you get a value that's much lower, and that's at 0.182 milligrams per liter, and that's a more accurate representation of the mean concentrations in that river. So we took a detailed look at our nutrient data from that multi-agency database, and we had about 25 million nutrient records. Of those, 60%, or about 14.5 million, had missing or ambiguous information for one or more of these key metadata elements that we needed to be able to confidently use the data. Some of the biggest offenders were knowing whether the sample was filtered or not, what the chemical form was, and even with units, there were times where we just didn't know whether, for example, it was milligrams per liter or micrograms per liter. If you are interested in knowing more detail about the metadata side of our study, there is a citation at the bottom, and it's also going to repeat later on in the presentation that you might want to check out. When we looked at the economic valuation of this ambiguous nutrient data that had been collected, it was estimated to come out to around $12 billion, and that's the amount that the originating organization spent to collect the data. And of course, this data was, in every case, successfully used by the originating data producers. So it met the original goals of data collection. The challenges come in for secondary use of the data when secondary users take the data and try to add value to it by using it for other purposes. And these challenges come in when data from one organization is combined with data from another organization that might be doing things differently. So the way to protect that investment and add value to that data to allow it to be used for a lot of different purposes and lead to a lot of additional insights is to implement standardized metadata throughout the United States. And there are efforts underway. Some of you may be familiar with WQX, those metadata standards, to help data producers get their data into a standardized format. And of course, the scientific value of metadata is that when you combine data from multiple sources, like we did in this study, it helps counterbalance the diminishing resources for stream monitoring within an individual organization. And it can also help lead to important regional and national insights that would not otherwise be possible. The practical challenge, of course, is that metadata harmonization is expensive, it takes a lot of time, and it's inexact. So there are these ambiguities in the data that data users have to make an assumption about if they're going to use it. And different data users may make different assumptions about the same data and could potentially reach different conclusions about important environmental issues. So once we had addressed these metadata issues and successfully harmonized the data that we were able to unambiguously interpret, the next step for us was to make sure that we had adequate data coverage to do a suitable trend analysis. As an example of the issues that we looked at in our screening process, this is total phosphorus concentrations in Fano Creek in Oregon. This is an urban stream where, somewhat unusually, they have a lot of phosphorus that comes in through groundwater. So when it rains and discharge increases, it actually has a dilution effect on the phosphorus in the river. So as discharge increases, you will see concentrations decreasing as that dilution increases. But there's a certain point at which there's so much rain and stream flow increases so much that you get enough runoff from the land surface that you start to see increasing phosphorus concentrations with increasing discharge. So when we do a trend analysis, we want to make sure that we have data from all across the discharge spectrum, so that we have low flows, we have moderate flows, and we have high flows, so that the resulting trend analysis is not biased to represent, for example, changes that are only occurring during low flow. We want to understand changes that are occurring on the whole at each of these sites. Similarly, concentrations will change seasonally. So here in Fano Creek, phosphorus concentrations tend to be highest in the summer, and then they decrease during the fall and the winter, and that has to do with the cycles of the wet and dry season in that part of the country. So we looked for sites that had data at least quarterly, so we could make sure we were capturing that seasonal variability, and so that we weren't seeing trends that, for example, only represented changes during the winter or only represented changes during the summer. We wanted to make sure we were reflecting conditions throughout the entire year. So now going back to this map we looked at earlier, where we had over 100,000 sites with one or more samples, and then I think it went down to about 80,000 sites when we had complete metadata. Now we're looking at 1,500 sites where we had start and end years that were within 1 year of our target start and end years for our 4 trend periods, and that had at least quarterly sampling. The final major step of our processing and screening was to obtain Streamflow data for these water quality sites. And I want to talk about the importance of Streamflow data for trend analysis. This is an example that is showing annual nitrate load to the Gulf of Mexico from the Mississippi and Atchafalaya rivers. And you can see the loads are shown here from 1980 to 2016. They vary pretty dramatically from year to year. And right now they have a reduction target of about 20% that I'm showing here in red. And you can see that there are some years where the load to the Gulf of Mexico is below the reduction target. So, you know, these are years where people tend to get really excited and think, hey, we're making a lot of progress, things are getting better. But in fact, when you superimpose here the annual pattern in Streamflow, you can see that every year that there is a low load, there is also a low stream flow, and that's because there's just less water in the rivers in the Mississippi River Basin to be carrying nutrients down to the Gulf of Mexico. So this is natural variability that is distinct from the deliberate, purposeful actions that we're taking in the watershed to try to manage nutrient transport. So in our trend analyses, we have information that is flow normalized. So it gives a picture of changes over time that are independent of these natural variations in stream flow and climate. So it's a closer look at trends that are being caused by human actions on the landscape. And you can see by looking at this green flow normalized line that the year-to-year change in flow normalized load is much less variable, and we're seeing some gradual declines over this time period. Another thing that including stream flow helps us understand is whether changes in concentration are different at higher flows during storm events and surface runoff than during lower flow conditions where maybe you're seeing more input from groundwater inflow. So this figure is showing nitrate concentrations at the outlet of the Mississippi River where it enters the Gulf of Mexico between 1980 and 2012 at high discharge in May. During high stream flow, of course, surface runoff is the major source of nitrate to the river. And so this decline that we're seeing may be evidence that there's been some progress in reducing nitrate and surface runoff through management actions. In contrast, we've seen an increase in nitrate concentrations at low stream flow, and most of the water in the stream is coming from groundwater inflows. So this suggests that nitrate concentrations in groundwater may be increasing and contributing to increasing concentrations in the Mississippi River. And it's important to know this distinction because of the slow movement of nitrate through groundwater to rivers. That means that recent increases that we're seeing in low stream flows could be a reflection of fertilizer application and other land management practices from many years ago. So for that reason, the full effect of today's management practices, for example, may not be measurable in these rivers until many years into the future. So the final part of our screening was to look at this flow component. Where could we pair water quality sites with a co-located or a nearby stream gauge, and where did we have the coverage over that range of flow conditions that I talked about a few slides ago? And that left us with 299 sites that met the criteria for our trend analysis. Now, I do want to point out here that there is most definitely other valuable long-term data out there in other parts of the country. What we're showing here are data that we could readily find through the steps that we took to compile data, and that met the screening criteria for this particular study. So for example, if you change the period of record that you were looking at, and instead of 1972 to 2012, you wanted to look at 1980 to 2015, you might end up with a different universe of sites here. I'm going to tell you a little bit about our final trend sites and results. This map here is showing, for all the nutrients we looked at in all 4 of the trend periods, how many sites passed our screens. And so we had 762 sites. And you'll see, looking at the legend, that there's a nice mix here of organizations. So the black squares are USGS sites. And you can see right away that had we not reached out to these other organizations for their valuable long-term monitoring data, we would have had a lot of geographic gaps in our coverage, and it would have limited our ability to talk about national patterns in changing stream quality. We have a mix here of state monitoring agencies, of NGOs and volunteer groups, tribes. We have what we have on here called local, which are municipalities, wastewater treatment facilities, soil and water conservation groups, and that sort of thing. So there's just an indication of the diverse monitoring that's being conducted in the United States, and all of the fantastic data that's out there. If you take a closer look at the situation with our nutrient trend sites, this is now just looking at nitrate. So we go down from 762 sites to about 400 maximum. And the number of sites available and the coverage of sites varies between the 4 trend periods that we were looking at. So by far the most sites we have is during the most recent period between 2002 and 2012. If you go all the way back to the Clean Water Act in 1972, now we only have 46 sites where there's been relatively continuous long-term monitoring between 1972 and 2012. Of course, most of you know that the Clean Water Act motivated a lot of new monitoring back in the 70s and 80s, and a lot of that has dropped off since then, so we don't have a lot of sites that have continuous monitoring during that entire period. Among the more straightforward results that we have found is the story of Diazinon. It's an insecticide that was developed as a replacement for DDT, and it was pretty widely used in the 80s and the 90s until they discovered that it was causing potential neurological problems in children. So EPA reached a voluntary agreement with insecticide manufacturers to phase out residential uses of Diazinon, and residential uses made up about 75% of total use in the United States, with the other 25% being in agriculture. And you can see here that in every site where we had enough data to do trend analysis, we saw declining concentrations between 2000 and 2012, and that most likely is a direct result of that voluntary phase out of Diazinon. That's almost the only straightforward story we have amongst our results. This is now showing nitrate concentrations during that same period, 2002 to 2012. And now I actually need to take a little time to explain the symbols to you. Of course, like we looked at with Diazinon, the black arrows are down. Open and filled is a measure of the confidence that we have in the trend. So the filled triangles, we have higher confidence in the trend. The red arrows are upward trends. And then the yellow circles are those where there's relatively stable conditions. We weren't able to detect a strongly downward or a strongly upward trend at those sites. And you can see with nitrate that there's a real mix. There's a real mix of stable conditions, of upward trends, of downward trends, and it varies throughout the country. So this is a situation where we need to spend more time figuring out what's going on, what's driving these regional variations, what's driving the increases, what's driving the decreases, to help us understand the factors that are affecting stream quality in the United States. So I mentioned now some of the interpretation that we're starting to get into, and I want to tell you a little bit more about the questions that we're going to be looking to answer with these results. So among them, what are the major causes of trends in U.S. stream quality? Where have we seen the biggest changes? Which chemicals have changed the most? So have we seen places where sediment's increasing but phosphorus is decreasing, and why might that be happening? Have our investments in pollution control been effective? That's our ability to answer that question is really tied to information that we have nationwide on pollution control activities. So where there have been agricultural conservation practices put in place, where wastewater treatment upgrades have been put in place, where industrial controls have been put in place of emissions. And that often is poorly quantified. So we're working with some of our partner agencies to get more information on that, but it's unclear at this point how definitively we're going to be able to answer that question. What's been the relative role of climate versus purposeful land use and management changes? Now moving from statistical significance to environmental significance, where have these trends been large enough to potentially compromise the health of humans or aquatic life? Or the flip side of that, where have they been large enough to successfully remediate problems that may have been present at some earlier date? And then finally, we are planning to update these trends through 2017 over the next couple of years. I mentioned that we had an online interactive mapper where you can access the results, and I'm going to give you a quick demonstration of that right now. When you enter the mapper, you're going to see, of course, in the center part of the application, a map showing by default nutrient trends for total phosphorus concentrations between 2002 and 2012. There are standard web navigation tools here, so you can zoom in or you can zoom out, and you can pan around to get a closer look. Along the left-hand side here are choices that you can make to change the display on the map. You'll see in the first drop-down menu the major categories that we looked at, so pesticides, salinity, etc. So if we pick major ions, you can then choose among the major ions that we have here. So I'll take a look at chloride. And you can switch between concentration and load if you're more interested in ambient concentrations or loading to some kind of downstream receiving water. That might be of interest to you to flip those. And then we have our 4 time periods that you can just easily switch between. There are also additional resources along the top here. There's a set of frequently asked questions where you can go in and find out how to access the data that we use. Why isn't my site in here? What might the potential reasons be for that? What is flow normalization? A whole number of different resources are available in here. We have a list of our partner organizations, so those organizations for which we have 1 or more trends among our results, they are all listed here to acknowledge their efforts. There's a list of everything that we looked at here, and you can get more information on those by taking a look at that list. And there's a user guide here that will help if you have questions about how to navigate through the mapper. Once you're in the mapper, you can also select an individual site, and it will provide some additional information for you. So it will tell you the site name. This happens to be the White River at Hazleton in Indiana. It tells you who collected the data. In this case, it's the USGS, and we got the data from NWIS. You can look at graphs of the data. So here we're going to be showing you monitoring data, the raw monitoring data. We're going to be showing you some of the model diagnostics, and then we're going to be showing you some of the model predictions. So you can click on any of these to see a bigger version of the data graphs. There's also a trend results tab where you can go in and find information for all of the trends at that particular site. So you'll see it's going to give you the chloride trend for the map that we were just showing, as well as everything else that's available at this site. So there's a fair number of nutrients and major ions, sediment, DOC, alkalinity, some pesticides that are available at this site. The table will give you information on the trend likelihood, the magnitude of the trend in percent or in original units, which for chloride would be milligrams per liter. And then it gives you the confidence interval on the trend to help you understand the confidence we have in the trend estimate itself. There's an option here to download that table, so if you want to export it and use it for local purposes, you can click on this download button. We also have a print feature, so if you select this option, it's going to generate a PDF of the extent of the map that you have displayed when you make the request, and it gives you some information on where the data, where the map came from. It gives you scale, and it gives the access date and the URL for where you access those results. Okay. I wanted to end by giving you the URL to the mapper, and I believe Dawn's going to send this out in an email, follow-up email. I mentioned earlier the metadata report that was published in the Journal Water Research earlier this year, so you can access that if you want more information. And then all of the detail on our data processing, the statistical methods we use for trend analysis, and the basic trend results are available in a USGS Scientific Investigations report. You can also access that online, and at that URL you will also find some data releases where you can access the input data to our models and the output data, the results themselves. And finally, I just want to acknowledge the efforts of my team. This has been a 4-year labor of love for us, and it's really been the outcome of all of their hard work. So I wanted to make sure I took a minute to acknowledge all of their efforts. And with that, I'm happy to take any questions. Thank you for your attention.
Thank you so much, Lori, and my apologies for the mispronunciation.
That's all right.
So we do have a few questions, but I'll just remind folks that if you would like to ask Lori a question about anything that you heard in today's webinar, go ahead and type that into the question box that's part of your control panel, and we will fit in as many of them as we can today. And as a reminder, we are recording this webinar, and we'll make the recording available online shortly. And as Lori mentioned, the materials that were in the second to last slide, those links will be included in an email that you will get as a follow-up to the webinar. So you will get that this afternoon. So here's one of the easier questions, I think. The 4 time periods, were they subsets? As I know you had like a 72 to 2012, then an 82 to 2012. Was it 72 to 82, 82 to 92, or are they really 40 years, 30 years, 20 years? How did that, what were the time periods there?
Yes, they are actually 40 years, 30 years, 20 years, and 10 years. With one of our models, we use 3 different statistical models, but the model we use for almost all of our data, you do have the ability to go in and make an estimate for any 2 years of your choice between 1972 and 2012. So if you were really interested in that, you could go download our results and make that calculation for yourself. We wanted to focus on those 4 periods, the 40 year, the 30 year, the 20 year, and the 10, because we had to pick something, first of all, and they provided some important temporal benchmarks. So 72, of course, the Clean Water Act. 1992 was the start of the NAWQA project, and then that 2002 period allowed us to look at the more recent period during which we know there's been a lot of emphasis on watershed management. So we were hoping to see some more recent information about the success or lack of success of those management actions.
Okay. Another question, and this is a little bit more technical one, so hopefully this will make sense as I read it to you. How do you handle censored trends, i.e. for less than detection limit when a trend observed when a lab measurement detection limit changes for a certain parameter? What % of the data needs to be censored in order to identify a trend as a censored trend?
Okay, well, I think that's a multi-part question. So first, we screened, this is a technical detail that I didn't get into in the presentation, but we wanted data sets that had at least 50% uncensored data for almost everything we looked at except pesticides. We allowed a little more censoring for pesticides because we wanted to get in some of those low-use pesticides. So with at least 50% uncensored observations, then the statistical methodology we used, I don't know if the person that asked this is going to be familiar with this, but we used a method that incorporates maximum likelihood estimation, and that is a way to handle censored data in a regression-based analysis. And maximum likelihood estimation can handle changing detection limits. So by minimizing the amount of censored data and using a method that allows us to accommodate more than one detection limit over time, we think we were able to successfully estimate these trends. Hopefully that answered the question. It's kind of hard when I can't talk to the person who asked the question, but I'm hoping that answered the question.
Okay, and if it didn't, Shibley,
email
please
me.
Feel free to email Lori. Her email's on the screen there. Okay, next question. How was data vetted for meeting QA, QC in collection and analysis? For example, here's an example. For example, did an organization have to have an approved QAP? Did the analysis have to be performed by an approved laboratory? Were methods of the analysis approved in 40 CFR Part 136?
Yeah, that's a great question. And the answer is no, no, and no. And that is not to suggest that we think there are no QA/QC issues in this enormous data set that we looked at. In fact, there almost certainly are QA/QC issues. The challenge that we ran into, and this is going to be a challenge that any secondary data user will encounter using data like this, is that we don't have ready access to information on QAPs, information on how a specific organization might vet their data before they make it publicly available. I mean, we're advocating as part of this process that the model that we eventually would like to see the monitoring community move toward is that if you're going to make data publicly available, that the data producers take the responsibility to make sure the publicly available data has been vetted, because they are absolutely in the best position to know where the problematic data are, to understand what the laboratory issues might be at their laboratory. It's very difficult for a secondary data user to understand those issues for a particular organization or to even have access to that information. So I think, what we had to do in order to make use of all of this information was assume that it was perfect quality data. Otherwise, we wouldn't have been able to use any of it. And so it was very much a trade-off between wanting to get that additional valuable information into this national evaluation and knowing that we were probably getting some small percentage of bad data. And so part of being transparent about that is inside of that mapper online showing the raw data so people can go in there for themselves and see whether there are just bizarre things that they think are in the data that might have affected our analysis. I will say that we did some very coarse screening, so looking for very unusual values that might indicate contamination or might indicate units problems, looking at the balance between total and dissolved and whether that was indicating a problem. But those are very coarse screens that aren't going to capture every problem. We did, I think, find a lot of problems when we checked the model diagnostics. So every model that we ran, we checked detailed diagnostics for them, and it took a long time. And there were definitely some wacky things that we found in the data by checking the model diagnostics, because the models just had such a tough time trying to fit a data set that clearly had some kind of QA/QC problem. So there were some vetting steps in there, but by no means, I think, did we capture all of the problems.
Okay, next question. Were all the sites included in this analysis on public land only? Or were there private lands, or I guess what kinds of, do you have that information?
I can't answer that definitively. We use sites that were in public databases. That's the only answer I can give. Based on the template that USGS uses for its NWIS database, if there are proprietary data based on where the data were collected on private land or near drinking water intakes or something like that, those sorts of data would not be included. But I don't know how other organizations handle that.
Okay. Next question, are the maps you developed available as web services? And if not, will you be creating web services in the future?
No, they are not currently. That's more of a resource constraint than anything else. We're talking a lot about future options for web reporting of trends, trying to do them on a more real-time basis, allowing more user control over the period of record, being able to calculate some of these trends on the fly. I think when we move to some of that, web services will be a more natural offshoot of how that technology is implemented. But there are some technical issues related to that, both on the IT side and also on the statistical side that we're working out. So it's probably going to be several years before that happens.
And a related question, are you thinking about adding seasonal trends to the maps?
So actually, and this applies to the last question too, all of our trend results are available through this link right here, through those data releases. So if you have a large volume need, you can, at a minimum, go there and download the results. Part of the information that's available there is going to be information that you can use to get seasonal trends, but it's less straightforward than just looking at the annual trend results. We do have the ability locally with some of the larger, more detailed output that we have from our models to do seasonal comparisons. And we are, for some of our interpretive reports, going to be looking at trends during different seasons. For example, some of our preliminary analyses suggest that if you look at chloride, which is an important consideration for corrosivity in drinking water supplies, that in the Northeast, we've been seeing increases in chloride concentrations, particularly in urban areas and particularly in the winter as compared to the summer. And there's some indication that that might be tied to increasing road salt use. So we do have the information available, but it's just hard to get through some of our reports. So if somebody has a particular interest in that, I'd encourage them to follow up with me directly.
Another question sort of about future work. This says, you described future work to identify potential causes of trends. Can you please elaborate on this, and in particular, what scale of analysis you are considering?
We're, at the moment, piloting a method that we've developed to do causal analysis using structural equation modeling. And very generally, that is going to be relating annual concentrations or annual loads and their changes over time to contemporaneous changes in potential causal factors. So fertilizer use, animal manure, human waste, atmospheric deposition, changing precipitation amounts, conservation practices, anything where we feel like they might be a potential causal factor, and we can actually generate a time series of geospatial data. So we're going to be using that information together with the annual time series from our models and concentration or load to try to pinpoint the major causes of trends. Now, the major causes of trends are going to vary, right, depending on which chemical you're looking at and which part of the country you're looking at. So part of the process of doing that analysis is identifying areas where we're seeing or we can hypothesize that there might be homogeneous causal factors. So looking at, for example, the Corn Belt, are there areas where we expect that some sort of agricultural change is going to be impacting water quality, and can we group those sites together to see if we're able to see a regional effect from that? We're not going to be doing causal analysis at individual sites for individual constituents because, as I mentioned, we have 20,000 trend results, so it's just really not feasible for us to do that many analyses and to eventually report them in any kind of coherent way.
Okay, next question. Are you going to be making any recommendations about how to standardize the collection of metadata based on the challenges you encountered?
Yes, so if you go to that report here, we talk a lot about the problems and make some initial recommendations, but I think the authority on this is not us, but can be tied to some of the efforts that are already underway to standardize metadata. And so one of the biggest efforts I would point to is through the EPA work group that involves states and other federal agencies to work on Storeit and the water quality portal, which combines data from Storeit and NWIS and other sources. Their WQX metadata schema is being updated right now, in part in direct response to this report, to enforce some standardization onto data that's being uploaded into Storeit and into the water quality portal. So we're trying to advocate for the use of WQX more broadly, either to have people upload their data into the water quality portal, which has the secondary benefit of making their data readily available in a centralized location that secondary data users know about and will go to, so that it will increase the use of their data. But they might also just want to implement those WQX standards in their own local database. The National Water Quality Monitoring Council has also done a fair amount of documentation of metadata standards, and they've really gone above and beyond the metadata elements that I talked about in this presentation, and talk a lot more about site location and laboratory method and all kinds of different things that people might want to check out also.
And that you touched a little bit on something in the next question. Are you planning on incorporating Storeit data into the analysis, as there is now a new combined USGS and EPA water quality portal?
Yes. Maybe that person missed the beginning of my talk. We did use the Storeit database for this current analysis. And yes, when we do the updates, we'll also continue to use both Storeit and NWIS data, in addition to all the other data sources that we looked at. And I'll give a shout out to the water quality portal that the person asking the question alluded to. It's a really nice centralized location that it's available online. It has a nice user interface that people can go in and easily draw data from both major databases in one location.
All right, next question. Are you using this data to report nationwide trends, even with large regional gaps, e.g. northern New England?
Yeah, anytime you do something like this where there are gaps, it's very difficult to say something that is defensibly, quote, national, right? Because there are gaps, there are regions that are underrepresented. We've been working a lot, for example, with our partners at EPA who do probabilistic sampling throughout the country. And for those of you who aren't familiar with that effort, they take the population of U.S. streams and they subsample from there, so that the streams that they are sampling can defensively be scaled up to make statements about national water quality. That type of study, because of the intensive spatial nature of it, we don't have the resources to collect, say, every month at all of those sites. It would be incredibly expensive. So they limit their sampling to a critical index period during the spring and summer at low flow. So they can answer one of their major questions, which is how do water chemistry stressors relate to aquatic life? And so by sampling during that index period, they're really able to answer that question in a way that is, again, defensively applied across the United States. In these kinds of data sets we're working with in our study, there are geographic gaps that prevent us from being able to make statements like, this is what's happening for the entire country. But these data sets are much more temporally detailed. They are continuously sampled over a long period of time. They are sampled under both low and high flow conditions. They are sampled throughout the year in different seasons. So we can say a lot more about the detailed changes over time, how things might be changing at high versus low flow, or in the winter versus the summer. And I've talked a little bit about how that information is important for understanding the causes of changes in water quality. So these two different kinds of sampling provide two different types of information. And I think right now we're working with EPA to show how they can be used together to provide complementary information, but they each have their own strengths and weaknesses.
All right, next question. Do you have plans to look at this data through a drinking water lens, meaning how the quality of drinking water sources are changing over time?
Yes, so right now we're working on a interpretation of these results where we compare the trends to drinking water standards. So, for example, at the start of the trend period, were concentrations generally below a drinking water standard, but by the end of the trend period, they were above a drinking water standard, pointing to a need to turn some attention to that site. And we also have the ability to link the location of our trend sites to the location of drinking water intakes, because some of these sites where we have monitoring data aren't close to a drinking water intake, so it doesn't matter all that much if the concentrations are above some drinking water standard. So we're comparing our trends to drinking water standards. We're also comparing them to aquatic life standards, and then taking into consideration where the location of the site actually is.
All right, I have a couple of questions specific to some of the examples that you provided. How do you think your phosphorus results compare with Stoddard's recent paper showing increases in phosphate across reference lakes and streams across the U.S.? What are the similarities and differences in your results compared with Stoddard?
So this goes back to I was talking about how we're working with EPA, and it specifically is linked to the work that underpins the study by John Stoddard. There they are seeing pretty widespread increases across the United States at more pristine areas that they think might be linked to an increase in dust and absorbed phosphorus. Our sites tend to be, and when I say our, I mean USGS and all of these targeted sites from different monitoring organizations, they tend to be located in areas where there's more development, closer to drinking water intakes or closer to agricultural areas or urban centers where we anticipate that there might be a problem that we need to track, or where we are spending money to clean up water pollution. So there's a specific purpose for monitoring there. So these results tend to be more directed toward those developed areas. We do have sites that are in undeveloped areas, and if you look at those, we're seeing a mix of increases and decreases just like we are everywhere else in the country in the more developed areas. And so this gets back to, and this is what we're trying to tease out together with EPA, is the Stoddard study was based on that probabilistic design where they were focused on low flow conditions in the spring and the summer, and in particular for this finding in more pristine areas. And so that's a particular set of information, and what does that tell us compared to what we're seeing in this data set with longer data sets, with higher flow and lower flow data, with data throughout all the different seasons. And what it's suggesting is that while we're seeing an increase across sites in the spring and the summer in pristine areas, that's not necessarily being carried over to sites in other developed areas and in other seasons and flow conditions.
Okay. I think we have time for maybe one more question here. Let's see. You had mentioned increased nitrate during dry periods in the Mississippi being due to increased nitrate in groundwater. Couldn't this also be due to the increased influence of wastewater during dry conditions?
Just to be clear, what I was showing there was the change in low flow conditions. So that's not necessarily saying just during droughts. It could be during an average flow year, but just during the low flow days during an average year. So times at which we think groundwater inflow is the predominant source. Yes, under those low flow conditions, point sources also could be a large source in certain parts of the watershed. Those influences tend to be fairly localized to the location of the discharge and some distance downstream in that tributary. By the time you get to that location at the mouth of the Mississippi River, the influence of point sources is most likely going to be outweighed by the influence of all of the other sources, including non-point urban inputs and in particular agricultural inputs. So we think that there may be some impact from point sources in that particular time series, but that most of it's probably going to be groundwater.
Okay, and for folks who may not have had their question answered, please feel free to email Lori directly and ask her if you have any additional questions about how these studies were conducted. And again, thank you so much to Lori Sprague for her time today in walking us through this. And again, the resources at the end of her presentation will come to you in an email this afternoon, and then as soon as the recording of the webinar is available, we will send you a link to that as well. Lori, any last words?
Thank you, everyone, for attending, and thank you for the great questions.
All right, everybody, have a great afternoon.